ranked the fourth most commonly valuable crop. Annual weight with Panama disease is one of the most devastating diseases that is caused by Fusarium oxysporum cubans. And these, among the tropical rainforest, is a particularly virulent strain that can infect 80% of the cultivar. And this disease can be found mostly in the major banana production area in the tropical and subtropical region. The effective and long-term management always challenges, largely due to the perennial host as well as the long-lived fungus spore in the soil. There are a lot of methods to control that, but they are not really effective and just slow the spread of a certain extent as well as have an impact on the environment. Therefore, the biological control using the microbe is considered to be the potential strategy. And the actinomyces is produced 20% the antibacterial as well as source to produce their secondary metabolite. And Streptomyces is one of the most important genus of actinomyces, can produce 80% of antibiotics as well as have capacity to produce the secondary metabolites. Therefore, we conduct our study on antagonistic properties against the Fusarium oxysporum cause disease on bananas of Streptomyces SVNCKH strain.
Our scientific research consists of three main contents. Firstly, for this scientific research, we conducted experiments to screening the antagonism of SVNCKH strain against Fusarium tropical race 4. After that, the inhibitory ability of the culture liquid on the growth of Fusarium tropical race 4 mycelium was detected. Drop the culture liquid from actinomycet cultured 7 days into a microscope slide containing developed Fusarium tropical race 4 mycelium. After growing at 28 Celsius degrees for 2 to 3 days, the mycelium samples were observed under the optical microscope. We also need to study on the effect of SVNCKH strain culture liquid on the Fusarium tropical race 4 spore germination. The experiment was conducted and the spore germination then was observed and numbered under microscope. After that, the cultured morphological characteristics of the SVNCKH strain were determined under ISP and Gauss type 1 media. Colony characteristics including vegetative hyphae, aerial hyphae colors, and soluble pigment production were evaluated. Nonetheless, the investigation of the influence of temperature, pH, and sodium chloride concentration on the growth and development of strain SVNCKH. Also, the strain identification was performed. In order to amplify non-ribosomal peptide synthase and polypeptide synthase, we used 8 different pairs of primers listed in the table below. Additionally, the data then is preliminarily processed on Excel software, RefPath Prism 9, one-way analysis of variance, and also Duncan's test. Based on those methods, we came up with the results. Firstly, we concluded the characteristics of our actinomyces strain according to observing aerial hyphae colors, vegetative hyphae colors, and soluble pigment production in the media system. Those results have been shown in the table. After knowing the colony's characteristics, SVNCKH actinomycet was studied to know the spore formation time, sporophore, and spore change shape. Observation results showed that the SVNCKH actinomycet strain started forming sporophores after 36 hours. The morphology of the SVNCKH spore chain is very characteristic, with a wide spiral, each chain forming 20 to 35 spores. Furthermore, the results after 7 days of culturing the SVNCKH actinomycet strain on the ISP6 medium illustrated that the color of the medium was changed from light yellow to black brown, proving that the SVNCKH strain can produce melanin pigment. We are also able to acquire favorable environmental conditions for SVNCKH strain growth and development. The results indicates that the SVNCKH actinomycet strain grows and develops well in conditions of temperature from 25 to 35 degrees Celsius, with a pH range from 9 to 6. SVNCKH is also able to develop a pH equal to 4, classifying this strain as a medium salt-tolerant group. For the next part, we have the classification results of SVNCKH actinomycet strain after using NCBI Blast and MegaX. Moreover, By studying the antagonism of VNO27 strain against Fusarium tropicoides 4, according to the growth inhibitor equation, the growth inhibitor value while using co-culture and agar diffusion method were 53.17% and 42.47% respectively. Furthermore, for the effects of VNO27 actinomycete culture liquid on Fusarium tropicoides 4 mycelium and spore formation, we observed that the mycelium in the experimental sample was abnormal. It appeared with protrusions and septum. Last but not least, by using 8 different pairs of primer, we are also able to amplify 5 genes encoded for polypeptide synthase and non-ribosomal peptide synthase, attributing to the secondary metabolites synthesis ability in VNO27 strain. Here is conclusion part for our scientific research. Thank you for your listening.